certain things are pretty important. But first of all, I'll put a few things down for short lecture. Meiosis. I know it sounds like mitosis, but it's different. Meiosis is sexual reproduction. So let's talk about here. Structure division. So I need to give you a couple of terms. We need to talk about haploid versus diploid. And then get into the details of I'm going to put a couple of notes up here. We'll start. So happens there are two meiotic divisions, and I'll get into all this. And so we call it meiosis 1, meiosis 2. Each one of these is, has a prophase, metaphase, anaphase, telophase. That's all the same idea, but we call it prophase 1, metaphase 1, anaphase 1, telophase 1. Then we call it prophase 2, metaphase 2, anaphase 2, telophase 2, depending on which meiotic division we're in. I'll come back to all that. I don't want to get into that right now, but just to begin with. So before I get into that, let me just give these words, haploid versus diploid. Then I want to say something to remind you about mitosis, what that's about, and then we'll talk about what we're talking about with meiosis. And why didn't they give names that were a lot different? I don't know. I can't tell you that. They just came up with these names. So we go with the names. But first of all, let me talk about this haploid versus diploid. When we're talking about eukaryotic cells, what do we know about the chromosomes for eukaryotic cells? They're in humans we have 46, right? But they come in pairs, right? Pairs of chromosomes. Eukaryotic cells have pairs of chromosomes. So humans have 46 chromosomes or 23 pairs, right? 2 number 1s, 2 number 2s, 2 number 3s, 2 number 4s, etc., right? 2 number 1, 2 number 2, 2 number 3, etc. All right.
Each being a set from each parent, right?
And one of each pair from each parent, yeah. One of each pair from each parent. So, each of us has 2 number 1 chromosomes. One of that came from our mom, one came from our dad. Each of us has 2 number 2 chromosomes. One from our mom, one from our dad. Okay. When a cell divides, normal mitosis and then cytokinesis, when a normal cell divides, what do we want? What do we want with mitosis? Back to the last lecture. Mitosis. God, it's been a long time, huh? Jeez. I kind of almost forgot where CR was located. It's been such a long time. No, I'm kidding. I actually went back to Washington, D.C. It was freezing back there. And then it got to be nice. But I got to see my grandkids. That was kind of cool. Little granddaughter, 3 months old. Anyway, that's another story. But, we're now back to this other stuff here, okay? Yeah. So when cells divide, skin cells are dividing all the time. Your bone marrow cells are dividing all the time. Various cells are dividing often. Some cells that we talk about don't divide. Some cells won't divide anymore. Some cells will divide if necessary, but they may not be dividing right at the moment. But the other cells are dividing all the time. And the ones that are dividing all the time go through mitosis. So mitosis, which we talked about last time, what is the idea of mitosis?
It's nuclear division of the cell.
Nuclear division. Now, so each cell, the cell that's going to divide, we call that the parent cell, is going to divide into 2 daughter cells. Yeah. But what do we want? That each daughter cell identical chromosomal information. Yeah. Yes. So mitosis, and I'll say followed by cytokinesis, of course. Okay, so mitosis followed by cytokinesis. The idea is that when a cell divides, when a cell divides, each daughter cell well, guess what? I never told you about haploid and diploid, which is what I said I was going to tell you. That's what I'm going to tell you right now. Each daughter cell has the identical chromosomal material of the parent cell. Has the same, we'll say, identical chromosomes as the parent. All right. Now, of course, in this case, we're just talking about one of your cells is a parent cell. It divides into 2. They're each the daughter cells of that cell. So we're not talking about, your parents and stuff like that. Although when we get to meiosis, we have to kind of think about that. But anyway, the point is that when a cell, like a skin cell, is dividing, you're supposed to end up with 2 number 1s, 2 number 2s, 2 number 3s that are identical to what the parent cell had, which means they're identical from what you inherited from your parents, okay? Whatever it was you inherited. Now, let's go back to this idea of 46 chromosome or 23 pairs. Because I want to use these terms, and we will be using these terms, in the future, so it's important that you get these terms. The terms haploid and diploid are just referring to the chromosome number. Now, with humans, when we use the letter N for the chromosome number, for humans, the 46 chromosomes, this is said to be the diploid state, is equal to diploid, and where 2N equals 46. So the total number of chromosomes is 46. N is how many different chromosomes you have, how many different kinds of chromosomes you have. So humans, so the haploid state, haploid for humans, this is for humans, haploid is 23. So we have 23 pairs, but what we're talking about is how many number 1s we have? 2, but so we only have 1, that we count. I don't want to say this. When I say how many different chromosomes, we have 2 number 1s. So better way to say this is this. Let's do this. Haploid is 23. And what we want is in what we call the gametes. Another word for gametes is germ cells. You can say sex cells, same thing. In the gametes, which is what we're talking about, eggs and sperm. In gametes, 23 chromosomes. haploid. This is for humans. It's for humans. For humans, you know, the gametes have 23 chromosomes. One number 1, one number 2, one number 1, one number 2, one number 3, etc. Okay. So the haploid state then for humans would be 23, because that's how many chromosomes are in a sperm or how many chromosomes are in an egg. But when a sperm and an egg unite, start forming a new individual, then what's that new individual going to have? 46 chromosomes.
So two.
Diploid, two in it.
Got it.
Does that make sense?
So the haploid is just the one set of parent
The
DNA.
one chromosome of each type.
Okay.
Diploid is when you have a pair of each type. Question?
So haploid is the number of pairs?
No. Oh, the number of pairs. Yeah.
And
Yes,
diploid
you
is
say
the
the number of
actual
pairs.
number.
Yes, the number of pairs. That would be true.
The haploid is the number of pairs, the diploid is the actual number.
And diploid is how many you actually have in most all of your cells. But let's just rather than saying that number of pairs is correct. That's not incorrect. What you said is correct. But when we really think about haploid diploid, this is what I want you to think about. I mean, nothing you said was wrong or anything else. I didn't say anything wrong. But think about it in terms of gametes. The gametes have 23 chromosomes, one of each type. And when an egg that's true for an egg, true for a sperm. When they unite, then you get a cell that has 46 chromosomes, a pair of each. Okay. So, if you take botany, if any of you go into botany, there's a bigger deal with that. With plants, they have these alternating generations where some of the plant exists as the haploid state, some of the plant cycle exists as the diploid state. I don't know much about it. I'm not a botanist, so I can't remember the details. I did know at one time all the details, but I've sort of forgotten it now. But anyway, but the point is that with animals, we're always diploid, except for the gametes, eggs and sperm. They're haploid. Okay. All right. So and if you think about it, to be honest, it was originally hypothesized that was probably the case. And then, remember I've said to you before that in science, if you can ask the right question, you might be able to get the answer. But so when they realized that the chromosome number was constant, so that you can look at any cell in a human, a skin cell or a liver cell or a brain cell or whatever, they all had 46 chromosomes. And they said, well, what about, if you're going to make eggs and sperm, if they both had 46 chromosomes and they joined together, then suddenly you'd have an organism with 46, 90, 92. You have 92 chromosomes. They said, then suddenly you'd be doubling all these chromosomes every generation. So someone hypothesized, over 100 years ago, that probably the eggs and sperm have only half the number of chromosomes, one of each pair. And so then they asked the right question. They could do experiments, staining techniques, and looking at it under the microscope. Yeah, that is the case. That the sperm and eggs only have one of each chromosome, one of each pair. Okay? How does that happen? Aha! That's the subject of this lecture. Meiosis. This is how it happens. Okay. So meiosis is a special kind of nuclear division followed by cytokinesis. So you remember we kind of get sloppy and just say cell division when we talk about meiosis and mitosis. We get sloppy and say cell division. Technically, it's nuclear division followed by cell division. So meiosis is a special kind of division process in which the number of chromosomes is reduced to half. And so it's called a reduction division, and that's what that whole thing means. So let me write that down in words so that we'll see it. So let's do it here. I'll say it. So what is it? So meiosis. What is meiosis then? Meiosis is a special type of nuclear division followed by cytokinesis, of course. So a special type of nuclear division for the formation of eggs and sperm, formation of gametes. Okay, no other kind of cell. Only when you're making eggs and sperm is there meiosis going on. So meiosis is a special type of nuclear division for the production of eggs and sperm. Okay. And it's called a reduction, and we'll say, and it is called a reduction division. Because the number of chromosomes is reduced in half. And that means that the original parent cell of where the cell is going to is going to form sperm, or the original parent cell is going to form eggs. That starts out with the normal 46 chromosomes. And it's going to go through actually 2 meiotic divisions, but in the process you're going to end up with 4 sperm, each one having 23 chromosomes. We're talking human, of course. Or 4 eggs, each having 23 chromosomes. One of each. I'll write that down. But eggs with humans and a lot of animals, it's a little different in terms of that, and I'll try to get into that in the lecture today. But just make it simple, not worry about the details. Okay, so let's see. So we'll say the original parent cell. I can't see this very well. The original parent cell has 23 chromosomes. No, sorry, 46 chromosomes. The original parent cell. I just forgot to write the word parent up there. The original parent cell is 46 chromosomes. But before you do any kind of division, what do you have to do? You have to go in S phase, back in S phase to duplicate all the genetic material, all the chromosomes. So the original cell has 46 chromosomes. Then in S phase of the cell cycle, in S phase, all the chromosomes are duplicated. I'm not going to give you any details right now. I'm just going to tell you the end process, and we're going to look at the details. All the chromosomes are duplicated. Then with meiosis, there are 2 meiotic divisions, which I mentioned over here. 2 meiotic divisions. Okay, the 2 meiotic divisions. And after the 2 meiotic divisions, from the one parent cell, we'll end up with 4 sperm, or potentially 4 eggs, each with halfway many chromosomes. One of each, number 1. One of each, number 2. One of each, number 3. Okay.
Why 2 and why 2?
Divisions? Or
Why
2?
1 egg or 4 eggs?
Oh, I'm going to show you. It will make sense. But let me write down. So the original cell has 46 chromosomes. Then in S phase, all the chromosomes are duplicated. Then after 2 meiotic divisions, meiosis 1, meiosis 2, after 2 meiotic divisions, 4 gametes are made. That's from the original cell. This is from the original cell, the original parent cell. 4 gametes are made. Each one is haploid. Oh dear, dear, dear. Okay. Now, I'll get your question.
Yeah, go ahead.
There's a lot of most of the events that we're going to talk about are very similar to mitosis, with a couple of major exceptions. But so the prophase, anaphase, metaphase, telophase are all very similar. But there's something going on here that requires two meiotic divisions, and I'm going to show you that right now. Yeah, and then I'll get to some of the details.
I think you're headed. I think you'll head to what I was asking.
Okay, let me try. Let me just erase this and let me show you what's happening here. So, maybe what I'll do is kind of compare mitosis and meiosis. Let's do that. I'm not going to do any of the fancy stuff. Just the simple stuff. As I said, there's a bunch of fancy stuff. Oh God, I tried to illustrate it in the lab today, and I was just having a horrible time doing it in the lab. But you're doing an exercise with pop beads. I don't know. Did any of you ever use pop beads when you were young? Make little bracelets or necklaces and little beads pop together.
Oh, those no.
My older daughter did that. Pop beads. Well, you're using pop beads in the lab to, sort of simulate chromosomes, and then you can do this event called crossing over. We're going to talk about it in a few minutes and all that. So and I should have just let the students do it with the pop beads instead of me trying to draw a picture because I had a terrible time doing it. So I'll do a better job tomorrow or Wednesday, I guess. So we'll see how. You got most of you are in Flet's class. So we'll see how she maybe could explain it better, or maybe she's using some fancy PowerPoint or some. I don't use those things. Anyway, I'm not out against it. I mean, I just don't like it. But anyway, okay. So let's look at meiosis versus mitosis. So let's go back to mitosis. And so we'll just do. Here's one cell. I'm going to do super simple, and we're going to say it has two chromosomes. It's got two. I should say, it's got say a little blue and a big blue, and a little red and a big red. Okay, so that's a big red and a little red. Okay, so it's got four chromosomes. It's got four chromosomes. Two number ones and two number twos. Four chromosomes to begin with. And then mitosis occurs. And you're going to end up with, well, remember what happened? Let's do S phase first. I shouldn't put mitosis just yet. I'll put mitosis down. So let's do S phase. What would we have then in our cell? Each chromosome was going to be made of two identical sister chromatids. So we're going to have here's one identical sister chromatid. Here's another identical sister, two identical sister chromatids. Here they are here. That's not so bad. Okay, so that's after S phase. Now, after mitosis, now we'll do mitosis. Now, I'm not going to do all the details of it. And mitosis followed by cytokinesis, of course. But I'm just going to say mitosis. And how about if I put it center, meaning for cytokinesis, so I don't have to write it down. So, in mitosis, then what are we going to end up with? Those two exact same cells that have,
one of each. One of each pair.
Yeah, a pair of each. That's right. So, two number ones, two number twos. Okay. Oops.
That got short. Yeah.
Okay, so now we have, it's just identical to this original guy. This is supposed to be a little bigger chromosome. Okay. But we had to, of course, duplicate all those chromosomes back in this phase to do this. Now, without going into any of the details of meiosis, we'll do the exact same thing with the exact same cell, but we're set to say it's going to make sperm and make eggs. So we'll still start out with the same parent cell. Here's the same parent cell. It's got a big blue chromosome, a little blue chromosome, a big red chromosome, a little red chromosome. Okay. Same chromosomes, same, two number ones, two number twos, one for the mom, one for the dad. And then it has to go through S phase. So same, everything's the same. S phase, everything is duplicated. And so now we're going to have Everything's going to be, each of these chromosomes is going to be made of two identical sister chromatids. I'm not going into any details right now, just giving you the overall thing.
Mm-hmm.
Okay. So I'm just going to show you the picture. We'll get back to it because there's more going on than I'm going to indicate. So we're going to have the first meiotic division. Meiosis 1. Now, after meiosis 1, what we get is one of each chromosome. So one big blue, one big, in one, well, who knows which one. Let's just, there's several possibilities, but I'm just going to show you what I'm going to do. What I'm going to do, we're going to have a big blue in this one, and a big blue, and a little blue in this one. Maybe a little red in this one, maybe, and a big red in this one. So this is after meiosis 1. We now have two cells. Those aren't eggs or sperm yet. We have two cells, and they have how many chromosomes? Only two. One number 1, that's the big one, and one number 2, the little one. I happen to put a blue one in this and a red one in this. Who knows which could go whichever way, but whatever. But one of the big ones, red or blue, and one of the little ones, red or blue. But two cells, two separate cells. Each of these now undergoes one more meiotic division. But notice we went from 4 chromosomes to 2 chromosomes. This is the reduction division. Now each of those chromosomes is still made of some sister chromatids. And in my drawing, you say they're identical, aren't they? It turns out they're not identical for reasons we're going to get to. But for right now, you can say, yeah, they're still made of sister chromatids. And we'll talk about the identical part in a few minutes, okay? You see what I'm saying? What we did is we separated the number 1 from the mom from the number 1 from the dad. We separated them in 2 separate cells now. Although there really is some combinations of 2, but whatever. Now each of these undergoes a meiotic division. The second meiotic division. So this is going to be meiosis 2. And what we're doing there is separating the sister chromatids from each other. So now we'll end up with 4 cells, 4 gametes. Now these are going to be the gametes. Maybe the eggs or the sperm, whichever. It doesn't really matter. But these are going to be the gametes. So I'll have you label these as the gametes. All 4 of those. But what we'll have is one number 1 and one number 2 in each cell. So that the way I've drawn it, I'll get a big blue, small blue, small blue, small red, big red, big red. So we end up with 4 gametes, each one with one number 1 chromosome and one number 2 chromosome. What did we get when we did regular old mitosis and cell division? We still had 4 chromosomes, but when we made gametes, 2 chromosomes. Do you see what I'm doing? Does it make sense?
Mm-hmm.
Or am I totally
Nope.
confusing you? God, I was droning on in the lab this morning. As soon as we were like, won't we ever quit? I finally quit. They were so confused I didn't know what to say. But anyway, but this is a little better. I think if you see this, I don't want to go into all the details because we'll run out of time and then you'll get all confused and bored and everything else. But I don't want you to get bored. I want you to understand the significance of meiosis. So starting out. Mitosis, we talked about last lecture, 100 years ago from before everyone to party. Where did you guys go? Nevada? You went to Nevada?
Yep.
I went to Washington, D.C. I didn't exactly party with my grandkids. But anyway, so that was a week ago. But whatever. We talked about mitosis, and that's going to be a skin cell. It could be, whatever. It could be a lining of your gut. One of those cells could be something from the hair. But the cells, start out with 4 chromosomes. When it divides, you end up with 2 daughter cells, each with 4 chromosomes. 2 number 1s, 2 number 2s for this. The original one had 2 number 1s. There's the blue and the red. 2 number 2s, the small blue and red. offspring cells, and daughter cells, same thing. Meiosis, you start out with the same kind of thing as this, as far as what the chromosomes, two number ones, two number twos. In S phase, everything's duplicated, same way as they were here. Everything's duplicated, identical sister chromosomes. After the division, there's some special things going on there, but after the division, we're going to end up with one number one chromosome and one number two in each of the two daughter cells. Each of those number ones, like this number one right here, is still going to be made of two sister chromatids. Here, this one, two sister chromatids, same thing here. So then they go through that second meiotic division, and the sister chromatids are pulled apart. So you end up with a gamete with one number one, one number two. Each one has one number one, one number two. Does that make sense? The tricky thing is, how do you get that actual reduction going on? How do you actually separate these? And this is where it gets, it's not super tricky, but it's just a little different from what we've learned from mitosis. But let's talk about it. In these divisions, the meiosis, there are two meiotic divisions, and each of them has a prophase, metaphase, anaphase, telophase, exactly the same words. The same basic things happen. Spindle fiber forms, the nuclear membrane disappears, the chromosomes become visible. All that stuff is pretty much the same thing, with one big difference in meiosis one. But let me just say, I can erase the mitosis thing here. I think I'll keep this meiosis little chart here because that'll help you remember what's happening. I'm going to erase this, but let me just, in terms of words, just so you get the terminology, and it's critical to get the terminology. Oops. So if you know about meiosis 1, there's a prophase 1, metaphase 1,
anaphase, and telophase.
And a telophase 1. I'm just using words here. I'm not going to go through all the details. And then, of course, there's cytokinesis. They don't say one or two of them. Then you have meiosis 2. And, I don't know the genesis. I need to write down what I'm doing here. I'm just telling you, just in terms of words, it's going to have the stages of prophase 2, metaphase 2, anaphase 2, and telophase 2. Then that's followed by cytokinesis. But we haven't gone into any of the details yet, but you can see what's happening in terms of this. After meiosis 1, we have 2 cells, but then each of those undergoes meiosis 2. We have the 4 cells, or 4 gametes, that's what we call them. They're still cells. A gamete is a cell. But I just want to keep reminding you that meiosis is just for the production of gametes, just for the production of sperm and eggs. Okay, so far? And in prophase, both of these prophases, the same kind of things we learned about the nuclear membrane disappears, the chromosomes become visible, the spindle apparatus forms, and they join with the chromosomes. But there's a real big difference here. So let me write that prophase 1. So we're in meiosis 1. Sometimes I put the Roman numerals kind of crossing, sometimes I don't. They are really Roman numerals, but it doesn't really matter. So now we'll have prophase 1. I'll just write those things down that we talked about before. Okay, the nuclear membrane disappears. Same as what we've had before for mitosis. Spindle apparatus forms. It's all right. I'll say something else. Spindle apparatus forms, and the chromosomes become visible. They supercoil. Chromosomes become visible. Now, what's each chromosome made of? Two identical
sister
sister
chromatids.
chromatids. Okay, let's remind ourselves of this. Chromosomes. Each made of two identical sister chromatids. Now here's the big difference. Okay, so that's all similar to what we talked about in mitosis, but now the big difference is this. Homologous chromosomes join. What do I mean? Remember the term homologous chromosomes? The two number ones are homologous, the two number twos are homologous, the two number threes. Let's get that term. Let's make sure that you have that term in your mind. Because otherwise it's going to make you know what's the term. So let's do this.
Instructions for the same thing.
So eukaryotic cells, the same, what I had before. Eukaryotic cells are contain pairs of chromosomes, right? Contain pairs of chromosomes, one from each parent. One from each parent. So we said two number ones, two number two, etc. Right? In this drawing, we only have two number ones and two number twos. I don't just go any higher. Obviously, in our cells, we have two number threes, two number fours, two number fives, all the way up to 23. The two number one chromosomes. Oh no, let's do it this way. Each, I better. This is true for the two number ones, true for the two number twos, true for the two number threes. So we'll say each pair of chromosomes is said to be, each pair of chromosomes is said to be homologous chromosomes. The number one from your mom, the number one from your dad, they're homologous chromosomes because they have the same genes on them in the same order. Are the genes from your mom and the genes from your dad identical?
No.
Not identical. There's going to be some variation. But they're the same genes. If you have a gene for eye color, the first gene on this chromosome, number one, it'll be the exact same thing over here. The first gene on your chromosome, number one from your other parent, as a gene for eye color. Maybe one's for blue eyes, one's for brown eyes, but it's still the same gene in the same location.
And the same type of information.
So it's the same kind of information. So the two number one, so each pair is of chromosomes is said to be homologous. So each pair of chromosomes is said to be homologous. So the two number ones, number one from mom, number one from dad, those are homologous chromosomes. The number 5 from your mom, the number 5 from dad, all of those are said to be homologous chromosomes. Now, what's the nature of our chromosomes right now, or just at the stage where we are right now? We've duplicated everything. So we have each chromosome is made of two identical sister chromatids. Okay, so here's number 1 from your mom, made of two identical sister chromatids. Here's the number 1 from your dad, made of two identical sister chromatids. Or my drawing up here, this would be the number 1 from this would be the number 1 from your mom, this would be the number 1 from your dad. Each one's made of two identical sister chromatids. Here's the important thing. Homologous, this only happens in meiosis 1, okay? In prophase 1. The homologous chromosomes join together. So I'm going to put it in red, because this is a big deal.
And that happens in prophase?
Only in prophase 1 of meiosis 1. The homologous chromosomes join together. Now, each chromosome is made of two identical sister chromatids. So here's the number one from your mom, here's the number one from your dad. They join together. How many strands do we have? 4. Or 4, how many chromatids? 4 chromatids joined together. So they're said to be a tetrad. They form tetrads because you have 4 sister chromatids all joined together. And tetrad just means 4, right? So homologous chromosomes join together as tetrads. So in this drawing right here, if I had this drawing, what I would do in the prophase 1, and so I'm going to be using this little guy right here. Without trying to get fancy here. Oh God, I'm going to try to draw some pictures and I'm going to make a big mess of it. I'm going to try. But just right now we can do this. So we're in prophase 1. Geez, I can't read this up here. Do a very good job of writing this. Let's write it again. This is prophase 1 now of meiosis 1. Meiosis 1, and we're in prophase 1 of meiosis 1. So I'm going to show you these things lying the tetrads, and I'm going to use this cell. Here's the cell, and here's the number 1 for the mom, the number 1 for the dad, or whichever color is which, I don't know. But those guys are going to join up. Here's the number 2 for the mom and the number 2 for the dad. Those are going to join up. So it would look like this. You'd have the 2 blues joined up with the 2 reds. The 2 reds, those are sister chromatids. And the same thing would be true for the little guys. So this cell would have exactly 2 tetrads. The 2 number 1s, each number 1's made of 2 identical sister chromatids. The 2 number 1s of the mom, the 2 number 1s, the 2 sister chromatids of the mom, the 2 sister chromatids of the dad are all joined together. This is for chromosome 1. For chromosome 2, the 2 sister chromatids of the mom and the dad joined together. Tetrads. See what I'm saying? No, you don't. Okay, so let me get this in there. I'll try to make sense of this.
Can you define what homologous is?
Okay, so
you
it's
keep saying something is homologous, but I'm not understanding what that means.
the number 1 from your mom and the number 1 from your dad are said to be homologous because they have the same genes.
So it's having the same genes?
The same genes in the same order. Now, is the number 1 from your mom absolutely identical to the number 1 from your dad? Not identical because we have variations in our genes. But, you might have, maybe the first gene on chromosome number 1 might be for one of the enzymes of glycolysis. You'll have an enzyme, you'll have the gene for that in your mom's number 1, same location, gene for the enzyme from glycolysis in number 2. I mean, it's a gene to make that thing. Does that make sense?
Functionally the same, but specifically different.
Yeah, I mean, that's right. It's the same functional gene, boom, 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 in order, same location. Now chromosome number 2, totally different genes. But the chromosome from your mom and chromosome number 2 from mom is going to have a certain set of genes like that. So if you actually drew it, so let's say here's a chromosome number 1, and here's gene. These are going to be genes here. Okay, here's gene 1, here's gene 2, here's gene 3, here's gene 4, here's gene 5. This is going to be chromosome number 1. And on your from the other parent, it's going to be the exact same order. Chromosome 1, and gene 1, gene 2, gene 3, gene 4, gene 5. Now each chromosome might have maybe has 10,000 genes. That's a chromosome. Same order of them. Now, will the gene 1 of your mom and the gene 1 of your dad be the same? In many cases, they might be identical, but there is variation, so they'll be a little different. Now, what if we're talking about that small one over there? The small one over there. Let's take little guy here, and this little red one over here. We'll say this has maybe gene 45 and gene 46 and gene 47. This would have gene 45, 46, 47. See what I'm saying? And so because they have the same kinds of genes on them, these 2 genes, even though you got maybe the blue one from your mom and the red one from your dad, they have the same genes on them, so they're said to be homologous. Does that make sense? Same information, same genetic information on them.
So number one is like eye color. The second one be like dimples in your chin.
Yeah.
The one after that, they're homologous because they have the same information.
Exactly, same information. You know, little variants of the information. That's why, you know, you might have brown eyes, and, you know, your brother might have blue eyes and something else. You know, variants, but still, it'd be a gene for eye color, a gene for a certain enzyme, you know, blah, blah, blah. So that's why I say when you have pairs of chromosomes, eukaryotic cells have pairs of chromosomes, and we number them just for convenience. You know, we have 23 chromosomes, but one of them is numbered chromosome number 1. But we have 2 of them, one from the mom, one from the dad. Those 2 number 1s are said to be homologous. The 2 number 2s, homologous. 2 number 3s, homologous. And what's amazing, and I don't know how they know this. Don't ask me how they know, but there's some way it's recognized that when you're in meiosis in prophase 1, the homologous chromosomes join. So here, this is the number 1, say, from your mom, the blue one maybe, and the red one's the number 1 from your dad. Now, you say, well, wait a minute, there are 2 strands there. Well, yeah, there are 2 blue strands and 2 red strands. The 2 blue strands because that number 1 was duplicated back in S phase, and they're identical sister chromatids. So the identical sister chromatids of dad's number 1, the identical sister chromatids of mom's number 1, join together. And so because you have 4 chromatids, you can count them there, 4. It's called a tetrad. Okay, does that make sense? There's one other big deal. And this gets to be confusing. I mean, it does get to be confusing. This is what I was having a heck of a time trying to draw in the lab today. The homologous chromosomes join, and this only all this special stuff I did in red here only happens in prophase 1 of meiosis 1. Everything else is pretty much like what we talked about in mitosis as far as what's happening. The homologous chromosomes join together as tetrads. Then crossing over occurs. Let me write the words and let me tell you what that means. Then crossing over occurs. Crossing over occurs between sister chromatids from opposite parents. I'll show you. This is the only time this happens. I'm going to redo my little. Instead of drawing them like little V's as I'm drawing them, I'm going to draw them as straight lines because it's a lot easier to draw them as straight lines. So I'm going to show you crossing over. So I'm going to take this particular tetrad. Okay, it's a tetrad. I want you to get familiar with the word. I'm going to take this tetrad. I'm going to just redraw it here. So I'll have here's 2 blue, the two big blues, 2 big reds. They're all joined together. This is the tetrad, right? This is a tetrad. Now your book, most books only show it happening between the close blue and the close red. In reality, it also happens between the outer red and outer blue too. I'm just going to show you with the close ones because I don't have to try to draw some of the pictures and mess up. But what happens, I'll show you. So I'm going to do this. This is going to be crossing over. So I'm going to redraw this. I think I can do it. See, I'm only going to do the near ones. So I'm going to do like this. And then I'm going to do like this. I'm going to do like this. Do like this. Okay. This is crossing over.
They're trading.
Let me write this down. Crossing over. And this is a special kind of thing. Crossing over. That one sister chromatid of the blue sister chromatid and the red sister chromatid, part of it crossed over. And then there are enzymes that will break the bonds between the blue and the 2 parts of the blue, and one part of the blue is going to join up with the red one, the other part of the red is going to join with the blue one, and you'll end up with, eventually, when they separate, which not immediately, but when they separate, it ended up like this. That's what it ended up. Can you see the difference between my blue and my red here?
Yeah.
I want you to see the difference between the blue and the red so you can see what happened. But the point is that
That's how they trade chromosomes?
so they're actually trading that double helix. You know, one double helix gets broken and joins this other double helix. Okay?
That's
I mean, it's kind of weird. Now, I showed you one crossing over event. In reality, in each, you know, those two little inner chromosomes, or the chromatids, I should say, there may be a dozen, maybe 15 or 18. Who knows? There's no rule. It's strictly random how many crossing over events occur. And supposedly, it always happens at least once. But it may happen a whole bunch of these crossing over events. And again, as I told you, most books don't show it. But the outer ones, this outer red one here, the outer blue one here, they can do crossing over too. There's a word for this in molecular biology. Let's write this word down. It's called recombination.
Will it trade out different homologous chromosomes in the same chain? Like, will it switch here and then down here switch?
Yeah, it can switch. So you could end up.
So to change the number ones
little blue,
and then.
little red, little blue, little red, little. I mean, yeah.
Wow.
It's gonna. So that's. I'm only showing you one place where it did it, but it can happen a dozen times, maybe 15 or 20 times. I don't know what the maximum number is.
23.
The longer the chromosomes, you know, because some chromosomes are long, some are shorter. The longer the chromosomes, the more chances for crossing over to occur, and it's random for where crossing over occurs. It's just a random thing, but it apparently always happens. They say. I can't guarantee that. That's what they say. It always happens. So what happens is there's actually a breakage of the blue chromosome right where they're crossing over, breakage of the red chromosome where they're crossing over, and then the blue one, this little piece of blue joins the red one, and the little piece of red joins the blue one, and you end up like that. Okay? This is said to be a recombination. What recombination refers to? When we're talking about genetics, you know, chromosomes and all that, what recombination means is the joining of DNA from two different sources. In this case, if this was meiosis happening in your cells, like you're producing sperm or you're producing eggs, we would be talking about maybe the blue chromosome maybe came from your dad and the red one came from your mom. Well, what we've done is we've actually broken off part of the mom's chromosome and joined it with the dad's, and broken off dad's and joined it with the mom's. So that these two inner ones are a combination. Like these two inner chromatids, they're not identical to your mom's genetic information or your dad's. They're a combination of the two. And it's a random combination. As I say, you're going to get more than just one crossing over event. You might get dozens. And as I said, books tend not to show you this, but you'll also get the outer chromosomes doing the same thing. So when I made this little simple picture down here with a big blue, a little red, a big blue, a little red, little blue, big red, little blue, big red, actually, each of these, like this big blue, would actually be a combination of blue and red. This little red here would be a combination of blue and red. How much of each? Depends on how many crossing over events. But every one of these will have a different sequence of these things because crossing over occurs, and it's not going to be the same in any two cells. So, you know, the number of possible combinations you can make is immense. You couldn't count them. I mean, because it's so wild. But let me answer questions, and then we'll just finish up on this. Go ahead.
Is that why when you have people that are related between generations, it varies so widely?
Oh, very, really widely. Yeah,
Like
exactly.
my sister and I, for instance, even though we look a hell of a lot alike,
Yeah.
it
But
could
there's-
be like a quarter different.
Oh, there's a whole bunch. You know, I mean, I mean, you'll see kids that look very much like one parent or the other, but still, there's going to be a lot of variability, and it's just because no two eggs or sperm are identical. You know, they just can't be because of all these random events that are occurring. And the more genes the more chromosomes you have, the more variability you're going to have anyway.
And at least your genes understand biodiversity.
But this is what This is a very important event because it adds variability to the gene pool. It adds variability to the genes in the pool of genes for that species, whatever the species, humans or whatever. Yeah, Megan.
So I can have very little expression of any of my dad's traits, but I can have a child that can have a lot of expression.
Good, that's right.
Of my dad's traits.
That's right, exactly. Yeah, I mean, and that's right. You never know. I mean, how it's going to come out until the experiment's done and you see, When we get to Mendelian genetics, which we're going to start, I think, next lecture. I think we're starting Mendelian genetics. Mendel dealt with these pea plants that only had 7 chromosomes, and he happened to pick some that worked out very nicely for him. Or it could be the case that he just said, if it wasn't working out nicely, he says, I can't understand this, I won't deal with it. That's a possibility too. But so he got these really interesting results, which started making people understand how genetics work. We'll get into all this next time. But the important thing here is that this is a very big deal because of providing variability in the gene pool. And that even though in my picture it looks like these 2 gametes would be identical and these 2 gametes would be identical, but actually because of crossing over, now they're going to be totally different. All right, so far now we've done. This is what I was having trouble is trying to draw these things up here. Maybe I could draw them if I go like this, if I keep it straight like this. If I go into, let's just go to metaphase. What happens in metaphase? They all line up at the
center
metaphasic plate, right?
Yeah.
Okay. So if I do that, I want, that if I put in my spindle apparatus, we'd have the spindle apparatus like this. You remember all that spindle apparatus and stuff like that. So I'll just try to do it very simply. So I'm going to do metaphase 1 here. Okay. And if I did that, and I'll just take this, I'll just make it this, and I'll do the same thing with the other 2. The little guys, the same kind of thing with the other 2 little guys. They all line up and they look something like this. I could do it. Let's see, I could do 1 big one, part of a blue, and then part of a blue. And the little one, I could do the same thing, a little bit. Now the red one would be, let's see, we'd have red here, red here, red here, red here, red here, and kind of put them apart here. Oh, I want a blue there. Dear, see how I get so confused. All right, it should look something like this. I oversimplify because I'm only having 1 crossing over event in each set of chromosomes. This is chromosome number 1 on the left. It's a tetrad. The 2 homologous chromosomes join together, but then we had crossing over. And then on the right hand side, that's chromosome number 2, say in my drawing. A little smaller chromosome. Again, just make it simple, 1 little crossing over event. But they're all lined up at the metaphasic plate. Now what's going to happen I'll get your question in 1 second. What's going to happen in anaphase? They're going to separate. Well, what's going to separate? Homologous chromosomes are going to separate. But the homologous chromosomes are no longer identical, are they? They're no longer identical because of
crossing over.
And the recombination, the crossing over, the recombination, so they're no longer identical. So that's a kind of a big deal. No question you have to play.
So the tetrads are lining up in this?
The tetrads then line up at the metaphasic plate. What lined up at the metaphasic plate in, I guess I erased mitosis.
In mitosis, yeah.
It was mitosis. It was just every single chromosome lined up in the metaphasic plate. They're only, but they're made of identical sister chromatids. So in mitosis, if we were talking about mitosis, what would line up would be all 4 of these guys would be lining up. All 4 things made of identical sister chromatids. Here we have only 2 things lining up because they're tetrad, because the homologous chromosomes join. So that's a big difference. So now in anaphase, let's see if we can kind of easily go to anaphase 1. Draw my circle. I don't want to put in all the spindle fibers. Let's just do this. We have blue, part of the blue. Down here we have a little bit of a blue. Over here we have, I kind of did the blues together tonight. Blue, a little bit of a blue. Down here we have blue. So let's see what we have here. We have red, 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 something like that. Oh, I didn't get to sort of like that. Did I do it right? Sort of like that. And then this is going to. These are going up to the top. These are going down to the bottom. So the homologous chromosomes separate. So let's actually write this down. So in anaphase 1, homologous chromosomes separate and go to opposite poles. But these homologous chromosomes, they're made of two sister chromatids, but they're no longer identical sister chromatids. So I'm going to put this up here. Hang on, what's it like? Made of two sister, I'll say each. Each is made of two sister chromatids, but no longer identical because of crossing over. And I should have written down one other thing here. Mike, go ahead.
You had mentioned that the other ones also.
Yeah, they do too. I didn't draw it in my drawing. Over here. See, the two inner ones did it, but I could also have it, this red one over here coming over all the way over, and the blue one coming all the way over. Okay. So they do too. As I say, most books don't bother with it, but that does happen. I should have said it metaphase 1. Let me just, since I didn't write it, but I should write it. It's in my drawing, but let's actually put it right down here. That the tetrads line up. Tetrads line up. That's a metaphasic plate. So now if you go back and review mitosis, you just have sister chromatids lining up, or each chromosome lining up made of identical sister chromatids. Boom, 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 boom. But here we have tetrads lining up. 4 sister chromatids, and they're going to separate. And so that's the actual reduction division. When you suddenly go from metaphase to anaphase, suddenly we're reducing the number of chromosomes in half because the original cell had 4 chromosomes, right? What's going to happen after this anaphase and then telophase? They get there and then it separates. You're going to have 1 chromosome, number 1, 1 chromosome, number 2. Now you're going to say, but each one's made of 2 sister chromatids. Yes, yes, yes. But still only 1 chromosome number 1, 1 chromosome number 2 instead of 2 of each. Okay. So this is the reduction division. So once we get to so we'll go through telophase, we'll go through cytokinesis, and then we'll have, and I'll write that down, but then what we'll have is 2 after meiosis 1, we'll have 2 separate cells with 1 chromosome number 1, 1 chromosome number 2. Each chromosome made of 2 sister chromatids, not identical the way they are in this picture. I told you I was going to oversimplify in this picture because we had complicated things to have talk about. There won't be identical sister chromatids, but there'll be 1 chromosome. In the next meiotic division, then these sister chromatids are going to separate into opposite cells, 2 different cells, and that's why you get 1 number 1 and 1 number 2. But of course, the number 1 won't be just blue; it'll be part blue and part red. This number red, this one right here, will be part red, part blue. It's going to be a combination because of crossover. So I oversimplified on purpose on that drawing because I hadn't talked about all that crossing over stuff. But the crossing over stuff is a big deal. It's a real big deal. And that's what makes meiosis so different and makes it so you get so much variability. So let's just put it in words then. I didn't want to go prophase, metaphase, anaphase, telophase, and get every single thing because we already talked about prophase over here, and then here's metaphase, they lined up. Here's anaphase, they separate. Telophase, they're going to get to the poles, and then cytokinesis, they separate. So let's see. Let's say okay. So then in telophase 1, chromosomes reach poles, then cytokinesis. But the chromosomes, what are the chromosomes made of? Chromosomes made of two not identical
Yeah.
sister chromatids.
So they're the recombined ones.
Say again?
They're recombined.
Because they're not identical because that crossing over and the recombination. Yeah, exactly. But that's prophase. I mean, that's all meiosis 1. What about meiosis 2 then? Meiosis 2, each of these cells, now we're going to have 2 cells, so now if I want to be a little more technical in my drawing here, I can do this, and it still won't be real accurate, but it'll be the right idea. I'm going to draw them straight across the way I did. Here's the blue, and then maybe this with some red. Here's a little blue, and a little blue here, and then here's red. I was going to do red here, like that, something like that. Did I do it? Sort of did it. Okay, but it's a combination of red and blue, even. This is supposed to be
But
number
there's
1,
still 4.
like that.
Ah.
And then so then down here it would be different too, because this may be this may be a little bit of red and a little bit of blue. I don't know. Whatever.
That is what it would be. And then
I'm not going to try to redraw all those, but you see what I'm trying to say. There's variability, but I made them in my original picture same for convenience sake, but in reality they're going to be a little different. So now what about meiosis 2 then? Meiosis 2. Oops, I didn't need to do that, did I? Oh, well, whatever. Let's say in meiosis 2.
So it has a similar prophase.
It's pretty much same as meiosis 2, very similar to mitosis. And the end result is that for each cell that was formed in meiosis 1, two gametes are formed. For each cell, there were 2 cells, right? We formed 2 cells. For each cell, very similar to mitosis. For each cell formed in meiosis 1, each cell formed in meiosis 1, 2 gametes are formed. And I would say parentheses, keeping in mind that, we're going to end up with a cell that contains, these 2 guys right here, and we're going to have, that doesn't have to go through S phase now, because we did the S phase way back. But we're going to separate the no longer identical sister chromatids. We're going to separate the sister chromatids. I don't want to draw that whole picture because it would take too long, but for each cell formed from meiosis 1, 2 gametes are formed, and in each case, the sister chromatids are separated. In each cell, the sister chromatids separate. We've already done the reduction division. So if I divide this cell in half after telophase and then cytokinesis, we've got one number 1 made of 2 sister chromatids, not identified anymore. One number 2. These guys are going to separate, so we get one number 1, number 2, in each one. Haploid. Each gamete is haploid. One number 1, one number 2. Am I making sense?
Yeah, it just seems like they're being divided a second time, one with half the information.
Yeah, exactly. It's going to have the number of chromosomes. They get one number 1, one number 2, one number 3. I mean, each gamete has one of each. But that is half the amount of genetic information. Absolutely. That's absolutely correct. Now, real quickly to say
But
something,
it's recombined
I
genetic
want to talk
information.
about just an interesting aspect of making sperm and making eggs. Because sperm are the smallest cells made, and eggs are the biggest ones. So, as long as you can kind of see what I'm saying here without going into all the detail, and you can look in books, diagrams in books and stuff like that, and I want it to make sense. On Wednesday, anything that doesn't make sense, I have to make sure it does make sense. Please try to understand this. The actual process of separating the chromosomes, it's all the same kind of stuff as mitosis. It's just the real big difference here. The real big difference is that in meiosis 1, the homologous chromosomes join, since each homologous chromosome is made of 2 identical sister chromatids, and 2 number 1s join together, you have a tetrad. 2 number 2s join together, you have a tetrad. The 2 number 3s join together, you have a tetrad. Okay? And then you get this crossing over, so you get all this variability. The rest of the process is more or less the same as what we saw with mitosis. I mean, it's basically the same idea. Just one thing I just want to tell you, this is sort of interesting, and for my purposes, it's not a big deal because I just want you to kind of see and this varies with the mammal, but I'm going to tell you about humans. With humans, all the eggs that are ever going to form in a human female are formed when she's a fetus. So all of you who are females, all the potential eggs you're ever going to form were established in the first couple of months of fetal development. Okay, so they're all there. Now, what happens is they start to go through meiosis 1, but they get into prophase 1 and they get locked in prophase 1. They don't go beyond prophase 1. So you have all these eggs. They're called primordial follicles. That's the egg plus some surrounding cells. But all these primordial follicles are locked in prophase 1. And then during the menstrual cycle, during the time when hormone from the pituitary, follicle stimulating hormone, it stimulates maybe a dozen or 2 dozen follicles to start developing. And what happens is those follicles start to develop, but the ovum, the egg part of the follicle, goes all the way through meiosis 1. This finishes meiosis 1. Now, most of those eggs die off. If the egg gets fertilized, then it goes through meiosis 2. The fertilized egg goes through meiosis 2. But in the process of doing this, you don't need to know this. I'm not going to ask it, not Sam, but it's kind of interesting. This would be the original parent cell, but then when it goes through meiosis 1, so this is to make eggs for egg formation, I should say. And these things like they call this the oogonium and all that. I don't want to get into all that. And the egg is the oocyte. I don't want to necessarily worry about it. But there's unequal cell division. So after meiosis 1, you get one big cell and a little tiny cell. Very unequal cell division. And then if it gets fertilized, then this will go into meiosis 2. And again, very unequal cell division. This little tiny cell. And this guy will go through. And these are called polar bodies. I'm not sure why they call them polar bodies, but I think having to do with the north and south pole on eggs when they're talking about it. Mm-hmm. But these are all called polar bodies, and they're nothing. They have the chromosomes, but nothing else. But by having this unequal division, you remember there was a lot of growth before this guy started this process. So it's a big cell. So you end up with one big egg. And all the others are just very small. But that's how the egg can be so big. It has almost all the cytoplasm, almost all the mitochondria, almost all everything. But only one set of chromosomes, just one number 1, one number 2, one number 3. And it's just chance which one it does it. When you're forming sperm, they get smaller and smaller. So with sperm production, you'd have this sperm production. They go through meiosis 1, and you still get 2 cells. Then these go through meiosis 2. meiosis two. This is meiosis one, sorry, meiosis two. But they get smaller and smaller and smaller. They start losing cytoplasm, losing everything, and then they start forming this little, just a little head with a little tail. So they're very tiny. But each one is functional. Whereas with the egg, only one becomes functional from the original parent cell. It's not a big deal for our purposes, but it's just kind of interesting. This is how you get the one big egg. And you've seen eggs before. I mean, look at a chicken egg, that's pretty big. Look at an ostrich egg, they're huge, you know. So eggs can be very, very large. They're the largest cells made. Sperm, smallest cell. And you know, from the point of view of forming a new individual, I mentioned this before, the sperm only gives chromosomes. It only gives genetic material. It doesn't even give any mitochondria. Yeah, there are mitochondria in the sperm. They don't get into the egg. Only so all the mitochondria that we inherit come from the law. It's just kind of an interesting phenomenon. So the sperm really, all they do are introducing DNA. So anyway, this is not a big deal for my purposes, but I just think it's kind of fascinating. Questions? Now I know that I didn't go through every single step here. I didn't really want to go through every single step, but I urge you to look at pictures in the book to make sure that it does make sense to you, and make sure you concentrate on these things I said were really important. Go ahead.
What happens wherever you get it?
Oh yeah, good point. The two different kinds of twins. What we call fraternal twins. That means two eggs formed. They both got fertilized. Separate sperm, separate eggs. So those fraternal twins are just like any normal sibling. They can be male, female, they can be male, male, but they're just like any normal siblings. With identical twins, once the egg joins with the sperm and it starts to grow and divide, if something happens and up to a certain stage they separate, then each one will grow into a separate individual, and they will have the exact same genetic information. You've heard of conjoined twins? Conjoined twins means that they're sharing something. That means they started to separate, but they didn't completely separate. And so whatever parts of the cells were stuck together, those stayed as part of one individual. There were other parts separated into separate individuals. So
So
they used to
cytokinesis
call those
broke at some
Siamese
point.
twins because 100 and some odd years ago there were these two conjoined twins from what we now call Thailand. In those days they called it Siam. And they were on display, so that's why they call them Siamese twins. The proper word is conjoined. But they just didn't separate. Weird. Yeah.